Have you ever looked in a mirror and wondered what truly dictates the speed at which we age? Is it just a random roll of the genetic dice? Or is there an actual master clock ticking away inside us? For decades, we've looked at wrinkles, gray hair, and stiff joints as the primary markers of getting older. But what if I told you that the ultimate pacemaker of human aging is hidden deep inside your chest, sitting quietly right above your heart? And what if I told you that for nearly a century, modern medicine completely dismissed this organ as a useless, vestigial remnant of childhood? It sounds wild, right? How can an entire organ be ignored by modern science? Before we uncover this fascinating biological mystery, a quick but critical reminder. This video is for informational purposes only and does not constitute medical advice. Please always consult a qualified healthcare professional for any health concerns or before exploring new health strategies. The frontier science we are discussing today involves experimental concepts that are still in early research stages and are not currently available as standard anti-aging therapies. Now, let's look at the clues. The organ I'm talking about is called the thymus gland. It is the primary training ground for your immune system, responsible for developing specialized white blood cells called T cells. But here is the bizarre twist. The thymus is the fastest aging organ in the vertebrate body. From the moment you hit puberty, it undergoes a progressive process called thymic involution, where functional tissue shrinks and is replaced by non-functional fat. By the time you reach age 40, your thymus produces only about 25% of the new T cells it did when you were a child. By age 65, that number plummets to a mere 10% because it decays so early in life. Classical biology assumed the adult thymus was just an empty shell. A Nobel laureate named Peter Medawar even dismissed it as an evolutionary accident of no very great significance. Doctors believed that once your initial pool of immune cells was established in youth, you could get by perfectly fine without the thymus. But as it turns out, this assumption was one of the biggest blind spots in medical history. The first major clue that shattered this myth came from the operating room. During open-heart surgeries, like bypasses or valve replacements, surgeons frequently remove the adult thymus simply to clear a path to the heart. Because the medical consensus stated the adult thymus was expendable, this was considered a harmless, standard practice. However, this practice accidentally created a massive group of tracking data. In a landmark retrospective study published in the New England Journal of Medicine, researchers evaluated the long-term health profiles of over 1,100 adults who had their thymus removed during surgery. They compared them to an identical group of demographically matched patients who underwent the exact same heart surgeries but kept their thymus glands intact. When the scientists looked at the 5-year post-operative data, they were absolutely stunned. The survival curves didn't just drift apart, they diverged violently. The all-cause mortality rate for the group that had their thymus removed was 8.1%, compared to just 2.8% in the control group. That means adults without a thymus had a nearly threefold increase in their relative risk of death within a 5-year window. But the surprises didn't stop there. The risk of developing cancer doubled in the group that lost their thymus, jumping to 7.4% compared to 3.7% in the control group. Even the incidence of autoimmune diseases saw a significant relative risk increase of 1.5 times under specific long-term analyses. When researchers performed deep blood profiling on these patients more than a decade after their surgeries, they discovered a permanent deficit in new T-cell production and a massive shift towards systemic, chronic inflammation. The evidence was undeniable. The adult thymus isn't a dead organ. It is an active, vital shield protecting us from mortality, cancer, and chronic illness. So, if losing your thymus accelerates health decline, what happens to everyday individuals whose thymus glands are just aging naturally? Does the speed at which your thymus decays dictate how fast the rest of your body breaks down? To answer this question, researchers turn to artificial intelligence. In prospective cohort studies published in Nature, investigators developed a deep learning framework to evaluate thousands of routine chest CT scans. The AI analyzed the size, shape, and degree of fatty degeneration of the thymus to generate a quantitative thymic health score. First, they applied this AI to a massive trial tracking over 25,000 asymptomatic adults over 12 years. The AI revealed a staggering correlation. Individuals with a high thymic health score enjoyed a 50% lower risk of premature all-cause mortality, a 63% lower risk of cardiovascular death, and a 36% lower risk of developing lung cancer compared to those with poor scores. Furthermore, a poor thymic score was heavily linked to systemic issues like metabolic syndrome, high blood inflammatory markers, and increased physical frailty. The AI also looked at a pan-cancer cohort of nearly 3,500 patients undergoing modern immunotherapy treatments. Here is what's fascinating. Modern cancer immunotherapies work by unleashing your body's existing immune cells to attack tumors. But the AI discovered that patients with robust, well-preserved thymus glands had a 37% lower risk of cancer progression and a 44% lower risk of death. Why? Because a healthy thymus provides a diverse, adaptable, and youthful immune system capable of fighting back. The researchers realized that the decline of the thymus isn't just a passive byproduct of getting older. It is a central pacemaker of systemic aging itself. When this gland degrades, your immune system loses its diversity, leading to a state of low-grade, chronic sterile inflammation across the entire body, a phenomenon scientists call inflammaging. This inflammation stiffens arteries, disrupts metabolism, and leaves us highly vulnerable to mutated cancer cells and novel pathogens. How do we stop this clock from ticking down? To fix the problem, scientists first had to figure out what triggers this rapid collapse of the thymic microenvironment. Research points to the structural collapse of thymic epithelial cells, or TCs, which serve as the physical scaffolding and training instructors for our immune cells. As we age, these master cells lose their ability to multiply, causing the entire structural niche to cave in. This decay is governed by a few fascinating molecular pathways. The sex steroid axis. The sudden surge of hormones like androgens and estrogens during puberty acts as a primary off switch for the thymus. Interestingly, experimental research shows that temporarily blocking these sex hormones can prompt the aged thymus to structurally regenerate, proving that the cellular machinery isn't completely broken, it's just switched off. The paracrine FGF21 mTOR pathway, a local metabolic hormone called fibroblast growth factor 21, or FGF21, has emerged as a crucial regulator. Advanced animal models show that when local FGF21 signaling is boosted, it activates specific intracellular pathways that preserve thymic cell count, increase volume, and directly reduce the accumulation of non-functional fat. The PLA2G7 pathway in diet. Data from human trials like the calorie study showed that a moderate 14% calorie restriction over 2 years significantly increased functional thymic volume and lowered fat buildup in middle-aged adults. Molecular mapping traced this effect to the inhibition of a gene called PLA2G7, which normalizes inflammation and protects the thymic environment from age-related lipoatrophy. But perhaps the most jaw-dropping clue comes from an entirely different species, the axolotl salamander. While humans have a very limited capacity to naturally repair damaged organs, the juvenile axolotl can completely regenerate its thymus from scratch after full surgical removal. Scientists mapping this process discovered that while a gene called FOXN1 is the master regulator for mammalian thymus development, the axolotl utilizes a primitive growth factor pathway called midkine, or MDK, to drive its miraculous regrowth. This ancient amphibian network is providing a literal evolutionary blueprint for human biotech firms attempting to stimulate entirely new organ growth in mammals. This brings us to the ultimate question. Can we use these molecular clues to regrow the thymus and potentially reverse aspects of human biological aging? Right now, a highly competitive biotechnology pipeline is racing to bring thymic rejuvenation out of the lab and into the clinic. Let's look at the four major strategies currently being tested. 1. Systemic endocrine restoration. The TRIM regimen, the pioneer of human clinical testing, is the TRIM and TRIMX trials, led by Dr. Gregory Fahey and biogerontologist Dr. Steve Horvath. In an initial pilot study, healthy men aged 51 to 65 underwent a 12-month personalized regimen combining recombinant human growth hormone, metformin, and DHEA. The growth hormone was used to directly stimulate thymic epithelial cell proliferation, while metformin and DHEA were added to protect against insulin-resistant side effects. The results were unprecedented. MRI scans confirmed a clear reduction in thymic fat and a restoration of functional, dense tissue. More shockingly, across multiple DNA methylation-based epigenetic clocks, the participants showed a mean biological age reduction of 1.5 years relative to their baseline, marking the first recorded reversal of epigenetic age in a human clinical trial. Phase 2 trials are currently underway to validate these findings across a broader demographic. 2. Targeted biologics and small molecules. Because systemic growth hormone therapy carries distinct metabolic risks, other firms are building tissue-specific tools. A startup called T-Cregen secured millions in seed funding to develop targeted thymopoietic biologics, growth factors designed to safely restore the thymic structure in situ without exposing the rest of the body to high hormone levels. Concurrently, a firm called Thymofox is designing oral small molecules to target dead zones in the aging thymus, attempting to flip the switch on resting age-associated epithelial cells so they can actively support immune cell maturation once again. 3. Reprogramming central immune tolerance. Instead of just boosting cell output, a company called ZagBio is utilizing the thymus as a therapeutic target to re-educate the immune system. Co-founded by Harvard immunologist Diane Mathis, they are engineering specialized antibodies to deliver specific autoantigens straight into the thymic medulla. The goal here is to leverage the organ's natural sorting machinery to eliminate autoreactive immune cells before they can cause harm, offering a potential breakthrough for preventing autoimmune diseases like type 1 diabetes without shutting down the patient's entire immune system. 4. De novo structural bioengineering. The most ambitious approach seeks to bypass our degraded biological remnants entirely. Thymic Therapeutics developed a machine learning platform capable of engineering brand new, functional human thymic epithelial cells from induced pluripotent stem cells. Their lead candidate demonstrated the ability to form a functional, vascularized neothymus in vivo within animal models. Recognizing the monumental implications of this technology, United Therapeutics Corporation acquired Thymic for a staggering $140 million upfront in cash, with hundreds of millions more tied to clinical milestones. They plan to utilize these bioengineered thymic grafts to establish long-term immune tolerance in complex organ transplants and combat general age-related immune exhaustion. Conclusion: the future of longevity. For nearly 100 years, science looked at the vanishing thymus gland and saw nothing more than a biological artifact, a useless piece of childhood scaffolding left behind by evolution. Today, we know the truth. The thymus is a foundational cornerstone of human health span, a brilliant shield protecting us from the slow burn of chronic inflammation, and a central pacemaker for how our bodies interact with time. While the therapies we've discussed are still navigating the rigorous path of clinical evaluation, the paradigm shift is already complete. We're moving away from simply managing the symptoms of old age and moving toward actively maintaining the youthful integrity of the systems that keep us alive. What do you think? Would you ever consider an experimental therapy to regrow a shrinking organ if it meant keeping your immune system young? Let me know your thoughts in the comments below. If you found this deep dive into the frontier of science fascinating, please hit that like button. Share this video with a friend who loves biology mysteries, and subscribe to the channel for more science-backed breakdowns. Thank you so much for watching, and I'll see you in the next revelation.